Thank you very much, Sandy, for this very kind, personal, and very generous introduction. It's a great pleasure to be here. I thank you and, of course, all the other organizers, Xiaowei, Atsushi, Rafa. It's, of course, a privilege to give this talk tonight and to tell you the story about super resolution and its basic principles. Now, we all know that throughout the 20th century, it was widely accepted, it was widely believed that the resolution of any light focusing microscope at the end of the day, for all practical purposes, is limited to about half the wavelength of light, or to be more precise, to a value of d, which is given by the wavelength divided by twice the numerical aperture of the objective lens. Now, this has been, so to speak, the general, basis of what people have saw that a light microscope is able to do. And, of course, many other techniques have been developed, as Sunny just mentioned, that have been, of course, very successful and very powerful, not at least the electron microscope. At the end of the 20th century, the gold standard in focusing light microscopy became the confocal microscope. And the confocal microscope was very important because it provided background suppression and it allowed us to do three-dimensional imaging non-invasively, especially in living cells. However, the spatial resolution of confocal microscopy was still limited by diffraction to typically about, say, 200, 300 nanometers. And so in this particular case, of course, we have an object, a set of proteins that it has been recorded confocally, but as you can see, it's not possible to see any details. Now, the development of STED microscopy at the turn of this century showed that you can really fundamentally overcome the diffraction barrier. There is land, so to speak, beyond the diffraction barrier. And so since we have developed this technique, I have now the option to show you an image that has been recorded with STED, and now you can see the details much, much better and see what it is. In fact, these are nuclear pore complexes from epithelial cell line of Xenopus laevis, and if you zoom in, of course, you see the details actually quite nicely. In fact, these nuclear pore complexes consist of 8 subunits, and why can you see them? Well, because the spatial resolution has been improved in this case over confocal by an order of magnitude. Now, an order of magnitude of resolution improvement really tells you a different story. That's very clear. I can't resist comparing it with the confocal microscope. And now you see that, this opens, of course, many, many possibilities. Now, as you may think, or you may think perhaps that in order to record such STED images, you require very complicated setups and systems. In fact, this recording was taken by a student in a student lab course setup. So if you think about that, then you will realize that the advancements that were actually made that has allowed us to get to this type of images cannot be technical or primarily technical. Of course, improved detectors, lasers, and so on played a role. But the advancement or the crossing of the diffraction barrier was certainly due to fundamental, say, insights, if you will, or very fundamental basic phenomena. And since I'm giving a keynote lecture today, I thought it's worthwhile speaking about these fundamental principles. And besides that, well, I've always seen this resolution question as a scientific quest. Is it possible to get beyond the diffraction barrier? And if it is, then why? What's the reason why we can get now these pictures, but maybe 20 years back we couldn't? This must have a very clear reason. I would like to talk about that, about the basic principles. Now, in order to explain the basic principles to you, of course, I'm coming back to very simple schemes. Now, this is just an object consisting of many molecules. These molecules are labeled since we do fluorescence microscopy. And what will happen if you shine light on them? They will be raised from the ground state to the excited state, and they come down, hopefully, by emitting a fluorescent photon, and that light is recorded by a detector or by the eye. Now, it's clear that we can separate features that are further away than the diffraction barrier. The problem of separation of the resolution exists only within a, if you will, diffraction limited range of around, say, 200 to 300 nanometers, as given by this equation coined by Ernst Abbe. In other words, if we manage to sort out the features within that range of about 200 to 300 nanometers, find a solution for separating, say, the strands residing here within this 200 nanometer range, we have sorted out the problem completely, because once you have a solution for that range, we can apply that to the rest of it. So we just concentrate on that, and so I can explain everything within this, say, typical range, because once I have a solution, I can apply it to the rest of the object, and we're done. So it's safe to concentrate just on that region and to ask, why can't we separate the features residing within this 200 nanometer zone? Well, one way of saying it is because we cannot concentrate the excitation light any further. If we could concentrate the excitation light further, as in near-field optics, we could concentrate, for example, the excitation light just on that strand, and then we could see that strand, and later on that strand, and so on. But in far-field microscopy, in a focusing light microscope, this is not possible. All of the features will be flooded at the same time with excitation light, and hence give off light at the same time, scatter light back, fluorescence light back, and so no detector will be able to tell these features apart. Now, if this is the problem, that all of them are flooded with excitation light, and all of them give light back simultaneously, a solution to the problem, of course, is to make sure that not all of the molecules that are flooded with excitation light, inevitably, are capable of sending light back. So in other words, you can say, well, we can't change the fact that all of them are flooded with excitation light. Maybe we can change the fact that all of them are sending light back. And if we can classify them, so to speak, some that send light back and some that don't, we can separate them. And this is actually the basic idea behind the STAD microscope. In a STAD microscope, you use a beam of light that shuts off molecules. So as you, perhaps all of you know, that beam is redshifted in wavelengths such that it instantly de-excites potentially excited molecules down to the ground state, just gets the molecules down to the ground state, and then they are not able to scatter fluorescent light back. So here, the molecules are flooded with a beam that is usually donut-shaped. We don't want to shut all of them off. We want to leave some molecules out. And now, those molecules are not capable of sending light back. They remain dark, while those in the center are still able to send light back, and so we can separate them and get a higher spatial resolution because we get just signal from those molecules. Now, why this would give us some improved spatial resolution, obviously, of course, we would like to do better. And this can be done, of course, by making sure that the molecules that reside also in here and in here are also silenced, are also shut off, not only those on the periphery. Now, what's the condition for that? The condition for that is easily found. In order to shut off a molecule by stimulated emission, not only you have to have the right wavelengths, you also have enough photons in the air. You have to have enough, say, photons that fall on a molecule, because once you have enough photons beyond a certain threshold, you can be sure there is always a photon, say, encountered by the molecule that kicks the molecule down in the case the molecule is excited. So once you pass a certain threshold, you can say you have switched the molecule off, and you have not allowed them to assume the excited state, and hence switch it off. You have deprived it of its ability to send light back. And so what we have to do in order to shut off also those molecules, it's very obvious. We have to increase the intensity of the donut such that the threshold moves in. And now also these molecules are turned off, and we get signal only from here. And this is what we want. Now we can separate signal from this strand from signal from the rest. But how we get the rest? Of course, we have to move the beam. Keep in mind, all of the molecules here are flooded with excitation light, but only these ones are allowed to emit. Those are silenced. And the rest, of course, we get just by moving the beam. As I said, now we get a signal from here, and then we get a signal in here. They are forced to emit sequentially in time because we play this on-off game. And this is how we separate the features and now can disentangle the features residing within this 200 nanometer range. Now, in fact, you can tune the spatial resolution, obviously, by moving the threshold in and out. If you reduce the intensity of the donut, it doesn't change in its shape, of course, but the threshold just moves in and out. Of course, then the resolution is poorer. If you increase it further, then the resolution will get up further. And the resolution is now not determined by Abbe's equation anymore. It can be described by a modified version of Abbe's equation. So what comes in is, of course, the threshold, because that tells us where the molecule turned off, and the total brightness of the donut. So the peak intensity is found in here. This is I, and the threshold intensity is 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 found in here. And the ratio of the two and the square root gives us this distance d, which eventually determines the spatial resolution. It gives us the region in which the molecules are allowed to emit, whereas in the rest of the area they are turned off. So one way of thinking about this, and if you have an optics background, you may be prone to thinking about that way, is to think, okay, this is non-linearity, what's going on here, because, say, having a threshold and shifting the threshold to the center is, if you will, a saturated transition that is taken advantage of in this particular case. I agree with that. But a word of warning, non-linearity is not the best line of thinking for very fundamental reasons. If you really want to understand these concepts properly, in all the ramification, of course, also the power and limitations, what we actually do is we transiently prepare the molecules in two distinct states. That's what we do. That's the only purpose of this beam. Keep it here in the ground state. Do not allow these molecules to assume the S1. Whereas in here, they are, so to speak, forced to assume the S1. So here then S0, here then S1. And because we prepare the molecules in two different states, we can separate them. Although all of them are flooded with excitation light, but they are in different states. They can send light back, but those can't send light back. And because these can send light back, we can separate them from the rest. That's the whole story in a nutshell. And as I said, we get the rest of the molecules just by moving this beam across the 200 nanometer zone. And of course, you always know where the signal comes from because that's preset actually by the zero of the donuts. That's predetermined. Okay, and so the signal here gives us, is proportional to the number of molecules that are found in here. So potentially many molecules that are in that range. Typically, there are many molecules. So how do we get the rest? Well, it's very simple. We could move the beam like that, and then you can imagine we get an image that is, say, 10 by 10 micron or whatever, or even larger. But of course, the larger the area gets, the longer it will take to take it in the image, because if you move a beam like that, the recording time scales with the area. That's very obvious. Is there a way to improve the recording speed? Yes, you can parallelize things. As I said, you need to sort out, the donor needs to sort out the features just within that 200 nanometer range. And so if we're having many of them to parallelize the imaging process, we can be much faster, as you see here, and hence record with a CCD camera or something. We don't have to worry that the signal from here is confounded with that from here, because it's further away than Abbe's diffraction barrier. So we need for the 200 nanometer zone only one, say donut, so to speak. They are sparse enough in order to be separated. The separation has to happen within a 200 nanometer range. Of course, parallelized, say, sub-diffraction resolution imaging was first done as structured illumination, pioneered by Mats Gustafsson and Rainer Heinzmann, and this is, of course, very well known as a concept. You can also use lines. They have to be further away than the diffraction barrier. If you use lines, you have to turn the lines to get also the resolution in the other direction. Now, in the beginning, of course, especially for STATS, since STATS requires relatively intense beams, we used single beams that were scanned like that, and including, let's say, the commercially available microscopes, so far use single beams. And I would like to show you a number of examples and of applications, quickly browse through them. Well, this is a relatively early one. So this is a protein called bruchpilot, was found actually by a colleague at the synapse, and he knew that it played an important role in structuring the synapse, but he didn't know how. So STAT microscopy showed for the first time that it forms these little baskets. Now, say, collaborating with him more recently, we labeled different proteins or different protein ends, and it was found, of course, you can find their, say, arrangement in space. It is a comparison with confocal after mathematical treatment, never gets as good as STAT after mathematical treatment, obviously. And so what you do is you can get actually from your data a three-dimensional model of the features in here, of the protein arrangement. And what I like as a physicist, that the numbers here are 30, 40, 50 nanometers, but the responsible wavelength is 750 nanometers. So it's a fraction of the wavelength. So although everything is here as a fraction of the wavelength, we can really tell it apart with focusing light, with far-field optics. This is taken from a Drosophila. So you can get a nanoscale arrangement of proteins at a synapse just by, say, overcoming the diffraction barrier. Now, this is another applicational study. HIV is typically about 140 nanometers in diameter, and an important protein on the HIV surface is this protein called ENF. It's found in about 7 to 14 clusters of proteins, but people had no clue how it's distributed on the HIV surface. Now, confocal microscopy, if you label it by an antibody and a dye, cannot resolve the pattern that may be there, and STAT clearly resolve the pattern. As you can see in here, and now you see that some of them have the ENFs all in a single place, some of them have patterns, and if you correlate it in space with the HIV, it was found that this ENF protein distribution strongly correlates with the maturation level of the HIV particle. To cut a long story short, it means that in order to be capable of infecting the next cell, the ends have to be more or less all in the same place, because otherwise it's not mature, not capable of doing that. So, a technical remark. Of course, different dyes have different excitation spectra, that's clear. And you know, of course, that you can excite with maybe one wavelength 2 different dyes. And the same applies in many cases actually to STAT. You can use one STAT wavelength to turn off several dyes if they're not spectrally too distinct. And this has been done in here. So here in this recording, we use just a single laser at 775 nanometer to silence 2 dyes. So in one case, you get something like 20 nanometer, the other one 30 nanometer spatial resolution. And because both dyes were silenced simultaneously, you have perfect colocalization, because the position of the emitter is imprinted by the donut, by the position of the 0. And so you have a perfect correlation in space. Another application, of course, for those that are interested in the neurophysiology. And of course, in the end, you want to see neurons in action in vivo, straight in a living system, in a living mouse, for example. And so here we used a transgenic mouse and provided a cranial window here by just opening the skull and focusing through a cover slip. And you see some of the neurons of this mouse expressed cytosolically the yellow fluorescent protein. And so we recorded in a STAT modality those neurons repeatedly. And you can see that this is taken on a minutes scale, that on that scale there are slight movements going on. So it's actually quite nice to see them. And this was only speculated before these results were made. Obviously, the brain is plastic, and the dendritic spines play a very, very important role. And these are postsynaptic sites, obviously. I think this kind of demonstration shows that there's a realistic chance to go into the brain and study the synapse here straight in the living mouse. And this is just a starting point. And so next step is, of course, to disentangle protein distribution and so on. This will certainly come. The spatial resolution is about 3 to 4 times better than what you have in the standard confocal or two-photon microscope. Another example. So this was recordings on the minute scale. Unfortunately, the room is not dim enough to see all the details in here, but doesn't matter. You still get the gist of it. Here, you see neuropeptide vesicles moving along motor neurons. Why I'm showing you this? Because it was recorded at 125 frames per second. So it's very fast recording. So the display is actually 4 times slower than the actual recording. It shows you can be fast with that and at the same time have an improvement of spatial resolution. Here in this case, it's about 70 nanometers. What's setting the limit to the speed? It's the brightness of the fluorophore in the end. The scanning can be made very fast, obviously. So this is an example of very fast scanning. If things become too fast, like molecules diffusing on the membrane, things go very quick, then you can say, okay, I do away with imaging and use STAT, for example, as a way to measure the diffusion very locally, say with high spatial definition, and see how molecules diffuse through the subdiffraction area. And this is called, for example, STAT fluorescence correlation spectroscopy if you combine it with STAT. But the advantage here is that you can probe much smaller scales. And for those of you interested in that area of application, the question is how small can you make that scale? And so in recent applications, we show that in artificial membranes, you get down to 15 nanometers in diameter. In live cell membranes, something like 20 nanometers. And this is, of course, quite a lot if you compare the diffraction resolution or the barriers set by the confocal microscope to about 250 nanometers. So there are many interesting applications you can imagine doing with this. It's an interesting alternative to using single molecule tracking, which is also very valuable. But the STAT FCS has the advantage that you can be faster. You can record faster, and you go straight to a certain area of interest. So talking about resolution, the question that comes up now, if one talks about the basic principles, what is the resolution that you can get? You've seen all kinds of numbers, 70, 50, 20, 15 nanometers. Well, it's clear that the resolution is determined, of course, by, say, if you will, the region, the coordinate in which the molecules is allowed to emit in this case. So we set up a difference in states, obviously. Here the molecules are allowed to emit. And so we can make the resolution very large by shutting off more molecules. And so D is, of course, given by this inverse square root law, I over IS. And you can imagine now that the